Student, in today's CET lecture, we are going to discuss the questions based on the Auxin and Gibberellin. In revision theory lecture, we have studied the all details about this Auxin and Gibberellin. So in this lecture, we are going to solve the questions based on the Auxin and Gibberellin. So whatever the question we are going to solve in this lecture, that questions already provided to you. You see that questions one by one. This question, it is taken from the one MCQ book. So as per the sequence given in that book, you see the questions. The first question in the sense, question number 156. See that question number 156, the Kogel H. Smith isolated dash dash from the human urine. Okay, scientists Kogel and S. Smith, they what they isolate from the human urine, that human being, they suffer from the pellagra disease. It is included in your textbook in introductory part. So with respect to that, options are given there. The first option, the naphthalene acetic acid. Second option, Auxin B. Third option, Auxin A. And fourth option, it is heteroauxin, or that heteroauxin, it is also called as a indole acetic acid. So I think you understand, from the urine of the human being, the scientists, the Kogel and Smith, they isolate the indole acetic acid in the sense heteroauxin. So the fourth option, it is your answer. Next question number 157. Chemically, heteroauxin is. आत्ताच आपण बघितलं की हेटेरोऑक्सिनलाच आपण इंडॉल ॲसिटिक ॲसिड असं म्हणतो. सो द केमिकली हेटेरोऑक्सिन इज द फर्स्ट ऑप्शन दे आर गिवन द इंडॉल ब्यूटिरिक ॲसिड, the second option naphthalene acetic acid, third option indole acetic acid, and fourth option it is the 2,4-D. So what is the correct answer? The heteroauxin means, just we have seen before, it is a indole acetic acid. So the third option, it is your answer. Then question number 158. Auxin were isolated by FW went from. Remember that Auxin, it was isolated by the scientist F.W. Wendt, and they performed one experiment there. That experiment, it is called as a Avena curvature test. So scientists F.W. Wendt, they utilize the Avena coleoptile tip, and from that Avena coleoptile tip, they isolate the Auxin. So with respect to this, there is a question, Auxin were isolated by Wendt from first option Avena coleoptile tip, second option coleoptile of canary grass, third option human urine, and fourth option corn gum oil. So what is the correct answer? The first option, it is your correct answer, Avena coleoptile tip. Then next question number 159. Auxin inhibit growth of. So always remember, the Auxin, they promote the apical dominance and they inhibit the growth of the lateral buds. So with respect to this, there is option. The first option terminal bud, second option lateral buds, third option foliar buds, and the fourth option, it is the stem buds. So Auxin inhibit the growth of lateral bud and they promote the growth of apical bud, in the sense they promote the apical dominance. Then question number 160. High concentration of synthetic Auxin means 2,4-D is used as first option the fertilizer, second option pesticide, third option weedicide, and the fourth option it is insecticide. So student, you are aware with the 2,4-D. It is always used for the control of the weeds. Okay, so it acts as a weedicide. So third option it is your correct answer. Then question number 161: the sprouting of potato during storage can be prevented by. So remember here, this question it is completely application based. Means how much you understand. It is checked with the help of a such type of the question. The sprouting of potato during storage can be prevented by. म्हणजे ज्यावेळेस बटाटे हे गोडाऊनमध्ये स्टोअर केले जातात किंवा आपण स्टोअर करतो गोडाऊनमध्ये असेल किंवा आपण घरामध्ये बटाटे आणल्यानंतर ज्यावेळेस ते एका ठिकाणी ठेवतो त्यावेळेस त्याला कोंब येतात, बरोबर, किंवा त्याला फुटवे फुटतात, त्याचे बर्ड स्प्राउट होतात, त्याचे डोळे तिथे ग्रो होताना दिसतात. तर हे जर आपल्याला टाळायचं असेल तर ते कसं टाळता येतं, कशाच्या मदतीने टाळता येतं, तर याच्यामध्ये ऑप्शन्स दिलेले आहेत. सायटोकायनिन, सेकंड ऑप्शन ऑक्झिन लाईक इंडॉल ॲस्टिक ॲसिड, अँड द थर्ड ऑप्शन जिबेरेलिन, फोर्थ ऑप्शन डी CU. ओके, दिस व्हॉट इज द करेक्ट आन्सर. तर बटाट्यामध्ये हे स्टोरेजमध्ये जे स्प्राउटिंग येतं ते स्प्राउटिंग आपल्याला अवॉइड करायचं असेल तर आपण त्याला कशाची ट्रीटमेंट देतो, तर आपण ऑक्झिन किंवा इंडॉल ॲस्टिक ॲसिडची ट्रीटमेंट देतो. तर इथं स्टुडंट तुमच्या लक्षात येते का की इंडॉल ॲसिटिक ॲसिड वापरून किंवा इंडॉल ॲस्टिक ॲसिडमुळं किंवा ऑक्झिनमुळं हे बटाट्याचे जे स्प्राउटिंग आहे ते का थांबतं. So what is meant by this potato? Let's see what is meant by the potato. See, suppose this is the ground level and suppose this is the potato plant. Remember this potato, okay, potato, it is a modified stem. बटाटा जे आहे ते रूपांतरित स्टेम आहे. So modified stem. In case of the potato, the axillary bud near the soil surface they sprout, and the growth of that axillary bud takes place in a downward direction. Also, such a type of a number of bud they sprout near the soil surface, and the growth of that bud takes place in downward direction, and the subapical portion of those buds they become swollen. This swollen portion of the lateral branch, that branch they grow downward. Such a swollen portions, these are called as the potato. Okay, these are called as a potato. So yeah, I think you understand how it is a modified stem. Okay, modified stem. Remember when we apply the Auxin promote the apical dominance and they suppress the growth of the lateral bud, in the sense that potato they are having the buds. Suppose this is a potato and this potato they are having the buds. These buds, all these buds of potato, these are the axillary buds. Okay, these are the axillary buds, and because that potato it is formed from the stem, and that stem they are having the nodes and internodes. Okay, they are having the nodes and internodes, and at the nodal region they are having a such type of the axillary buds. So when such a stem is converted into this potato, what happens? Whatever the buds present over the potato, all the buds are the axillary buds. So when we apply the Auxin at the time of storage to that potato, all this growth of the bud it is automatically checked, because this Auxin they suppress the growth of lateral bud and they only promote the growth of the apical bud. So the apical bud is not here. All the buds present over that it is the axillary buds. So because of this, for preventing the sprouting of potato during storage, what we apply? We apply the indole acetic acid. So, student, this is the question. It is totally based on your applied knowledge or allied knowledge. Okay. So, what is the answer? The second option is your answer. Auxin, like indole acetic acid हे वापरून आपण बटाट्यामधलं स्प्राउटिंग स्टोरेजच्या वेळेस आपण थांबवू शकतो. नेक्स्ट क्वेश्चन 162. The site of maximum synthesis of auxin is first option mature root, second option meristematic region of shoot, third option endosperm, fourth option leaves. So what is the site of maximum synthesis of auxin? It is the meristematic region of the shoot. So the second option, it is the correct answer. Then the next question, question number 163. Very high concentration of auxin. When there is a high concentration of auxin, what happens? First option inhibits flowering, promote flowering. Third option promote growth of stem. The fourth option inhibits growth or kills the plant. So when there is a maximum concentration, automatically they inhibit the growth or they kill that plant. Okay. So as per requirement, the auxin, they are there and they induce their effect. Then the next question, question number 164. The auxin play important role in first option flower initiation, second option prevention of abscission layer, third option parthenocarpy, and all of these. So the question, auxin play important role in what is the correct answer? All of these. Means they plays an important role in flower initiation, then prevention of the abscission layer, and also they induce the parthenocarpy. So all the role they perform. Then the next question, which of the following increases number of female flowers? So when the flower initiation, it is based on the concentration of the respective hormones. So with respect to that, I think we are discovered or we are discuss the role of the auxin and gibberellin with respect to the development of the type of flower, in the sense male flower or the female flower. So which with respect to this, there is a question, which of the following increases number of female flowers? The first option auxin, second option cytokinin, third option ethylene, and the fourth option it is all of these. So these three growth regulators, they take part in the formation of the female flowers. So all of these means fourth option, it is your correct answer. Then the question number 166. 2,4-dichloronisole means DCA is the first option auxin precursor, second option antioxin, third option bound auxin, and fourth option free auxin. So the correct answer is the bound auxin. So the 2,4-dichloroanisole means DCA. It is a bound auxin. So you remember, student, I think this is not given in our textbook, but for your information, you have to remember this. Then the question number 167. Increase in yield in plant like cucumber by application of auxin is due to. This question, it is also based on your application knowledge. Means when we apply the auxin to that cucumber, then that cucumber they have to develop or they develop the female flowers, and when they develop the female flower, number of female flowers are increases over that plant, then automatically the yield of that crop plant, it will be increased, because the female flower they take part in the development of the cucumber fruit. Okay, so with respect to this, there is a question: increase in yield in plant like cucumber. The application of Auxin is due to, कारण कुकुंबर प्लांट्स मध्ये काकडीचं उत्पादन हे वाढतं की ज्यावेळेस आपण ऑक्झिन स्प्रे करतो, कारण त्याच्यामुळे तिथे फिमेल फ्लावरचा नंबर वाढतो आणि त्याच्यामुळे उत्पादन वाढतं. हा तिथं क्वेश्चन होता. त्याच्यानंतर क्वेश्चन नंबर 168. The application of auxin causes formation of seedless fruit. It is called as, when there is a formation of seedless fruit, in the sense the fruit formation without fertilization, it is called as the parthenocarpy. So when we apply the auxin, we are to induce the parthenocarpy. Then the next question number 169. The elongation of stem is caused by, when there is a stem elongation, then who is responsible for the elongation of stem? The first option auxin, second option gibberellin, third option cytokinin, and fourth option both A and B. So elongation of stem, it is due to the application of the auxin and gibberellin. Means fourth option, it is your answer. They are given there, both A and B. Then the next question number 170. Which one of the following groups comprises synthetic auxin? So ऑप्शन्स मध्ये ग्रुप्स दिलेले आहेत आणि त्या ग्रुप्स मध्ये सगळे सिंथेटिक ऑक्सिन कोणत्या ग्रुप मध्ये आहे ते तुम्हाला ओळखायचं आहे. द फर्स्ट वन 2,4-D, 2,4,5-T and CRT. Okay, then the second group gibberellic acid, abscisic acid and indole acetic acid, and the third option it is 2,4-D, 2,4,5-T and naphthalene acetic acid. And the fourth option, it is the cytokinin, indole acetic acid, indole butyric acid. So what is the synthetic group? So in all total, the three hormones present in which group? And totally that three are synthetic one. So the third option, that is 2,4-D, 2,4,5-T and naphthalene acetic acid, this is the group of the synthetic auxin. So the third option, it is your answer. Then question number 171. Which horticultural application will auxin have with cuttings of rose and bougainvillea? ज्यावेळेस आपण गुलाबाच्या कटिंगला किंवा कागदी फूल की ज्याला आपण बौगनवेलिया म्हटलं जातं त्याच्या कटिंगला ऑक्सिन वापरतो, म्हणजे तिथं नक्की काय होतं हे विचारलेलं आहे. फर्स्ट ऑप्शन फ्रूट इनिशिएशन, सेकंड ऑप्शन रूट इनिशिएशन, थर्ड ऑप्शन पार्थेनोकार्पी अँड द फोर्थ ऑप्शन सेक्स एक्सप्रेशन. तर ज्यावेळेस आपण ऑक्सिन कटिंगला लावतो त्यावेळेस तिथे काय इंड्यूस होतं? रूट इनिशिएशन. सो द सेकंड ऑप्शन, इट इज युअर आन्सर. Then the next question, the question number 172. When ori is converted into fruit without occurrence of either pollination or fertilization, the process is called. So automatically, I think you understand well. Just before this, we solved one question. What do you mean by the parthenocarpy? So when instead of fertilization, when the ori they convert into fruit, it is called as the parthenocarpy. So it is the second option, it is your answer, parthenocarpy. Okay, then the question number 173. Which property is specifically attributed to gibberellin? Okay, which property is specifically attributed to gibberellin? The first option, apical bud growth initiation. Second option, root initiation in stem cutting. Third option, elongation of genetically dwarf plant. And fourth option acts as a selective weedicide. So student, what is the correct answer with respect to the gibberellin, the elongation of genetically dwarf plant. So when we treat the dwarf plant with the gibberellin, then that dwarf plant they grow as like the normal tall plant. Okay, so this is the answer. Then the next question, the question number 174. The gibberellin was first isolated from. So that gibberellin was first isolated from the first option given, fungus causing disease in rice seedling. Second option, root of diseased rice plant. Third option, bacteria which causes disease to rice plant. Fourth option, viruses causing disease to rice plant. So what is the correct answer? The fungus which causes diseases in case of the rice seedling. So the first option, it is the correct answer with respect to isolation of the gibberellin. The growth hormone responsible for bolting is. So what is that bolting? That bolting, it is seen in cabbage, beet. So in that we saw in the revision theory lecture. In that the bolting means because of in case of the cabbage. Okay, in case of the cabbage means it is the largest bud among the angiosperm. In the sense, in that cabbage, the internodal distance reduced, and all the young leaves they placed very closely, and they form a compact ball-like structure. So such type of the cabbage they show a such type of the compact ball-like structure. Okay, so such a type of the structure it is seen in case of the cabbage. But when we apply the GA, okay, when we apply the GA to such a cabbage, what happens? This internodal distance it is increases, and that cabbage it is converted into a tall plant. Okay, so such a type of the plant. So such a type, such type of the conversion, it is called as the bolting. Okay, so see the next question, the question number 177. The gibberellins were first discovered in 1926 by this. The name of scientists are given. The first option Cross, second option Gane, third option Kurosawa, and the fourth option it is the Darwin. So what is the correct answer? The Kurosawa, it is the correct answer. Gibberellins were first extracted from first option fungus, second option higher plants, third option algae, and fourth option it is moss. So they were first isolated from the fungus. See the student, last question from this lecture. The question number 179. The dash isolated the gibberellins from the culture of the filtrative fungus. The first option Kurosawa, second option Cross, third option Yokuta and Smoky, and the fourth option Darwin. So who isolate the gibberellin from fungus in crystalline form? It is the Yabuta and Smoky. So the third option, it is your correct answer. So student, with this we will stop at this moment. The remaining question, whatever the questions are remain, that questions you have to solve at home, because we have discovered this Auxin and Gibberellin in detail in our revision theory lecture. So when you read that questions, you can easily understand that question, and you can easily move towards the respective answer. Okay, so student, you have to solve all those questions at home. So with this we will stop at this moment.